When Harold Varmus, president of the Memorial Sloan Kettering Cancer Center in New York, was asked to speak at the Guggenheim Museum, he worried that a straight lecture might not fit in with the more artistically presented programs the Guggenheim's visitors were accustomed to. So he called his son Jacob Varmus, a musician and composer, and Genes and Jazz was born. With science content delivered by a Nobel Prize-winning researcher, music performed by some of New York's most sought-after and talented jazz musicians, and films by an award-winning biomedical animator, Genes and Jazz is truly a sensory celebration. Ladies and gentlemen, Genes and Jazz.
Good evening. I'm Harold Varmus.
And I'm Jacob Varmus.
Welcome to Genes and Jazz. For the past year and more, my son Jacob, the musician, and I, the scientist, have been working on a performance that we hope will illustrate the kinds of work we do in our individual lives and shows in which the ways in which the worlds that we inhabit can intersect.
I am not a scientist. Somebody asked me yesterday, this interviewer asked me, what does science mean to you? I mean, I guess it means the pursuit of knowledge, right? But I always had a hard time learning things by rote. I have a big problem with the way biology is taught in our classrooms. And I flunked high school biology. I got an A in summer school. Counts for something. But I was the kid whose chlorophyll calibration strip never turned green in the right place. And I wanted to do something with Harold, my father, to showcase his artistic side as well and create some musical forms and settings that are going to bring these fascinating subjects to life for all of you. We hope you enjoyed the show, and say it again.
Well, thanks for coming this evening. You will enjoy this, we hope, and we're going to try to have fun as well. Thank you. So this evening I'm going to tell you about cells, what they do, and how they know what to do, about that information and how it changes over eons to diversify life's forms, and during a single lifetime to cause a cancer. All of this, believe it or not, in 3 short acts: the cell, evolution, and cancer. We begin with this drawing of a cell, the fundamental unit of living things. A typical cell from animals like us is probably familiar to most of you. It has a nucleus, which is surrounded by a cytoplasm. The entire cell is held together by an outer membrane, the nucleus by an internal membrane. Cells are tiny orchestras. They contain several kinds of instruments. Chromosomes, which contain genes and reside in the nucleus. Enzymes, proteins that synthesize some molecules and destroy others, and are found everywhere in the cell. The membranes, which let chemicals in and out. Mitochondria, which generate chemical energy. And ribosomes, microscopic factories that make proteins. As in a musical orchestra, these instruments must harmonize. They're not playing solos. They have rhythm and dynamics, with different paces under different conditions. They play repetitive chords rather than distinctive melodies, and they are adapted to achieve the functional purposes of the entire cell. Cells use their instruments to create the harmonies of life. The human cell types on the screen behind me are united by their common instruments and harmonic rules, but at first you'll be more impressed by their differences in appearance and function. Skin cells form a barrier to infections. They produce hair, and they can reveal our ages. Red blood cells carry oxygen from our lungs to our tissues, and white blood cells respond to infectious invaders and can actually engulf them. The nerve cells in our brains allow us to think, to retain memories, to tell our muscles to contract, and to appreciate signals from the sensory organs so that we can see and touch, smell and hear. Other cells are connected to nerve cells in the brain and convey sensations to them. So when the musicians generate vibrations in air that move the antennae of your ear's hair cells, a signal flows to the brain's neurons so that we can hear and retain memories of sound. Other cells maintain our many kinds of tissues. Bones have cells that dismantle the hard part of bone and other cells that rebuild it. Our organs, the lungs, intestines, the pancreas, and endocrine glands, are rich with specialized cell types called epithelial cells that perform the functions of those organs. Most cancers, as you'll see, arise from these epithelial cells. Our germ cells, sperm and eggs, confuse to form the first cell of an embryo. And after a few divisions, stem cells can be derived from the new embryo. These cells can famously become any type of cell. Our bodies contain literally hundreds of specialized cell types, not just the 10 or so shown here. But for now, we're going to focus on some common properties. One feature is essential to all forms of life: the cell division cycle. Animal cells, plant cells, bacterial cells must all grow in size. They must duplicate their many components and then separate into two daughter cells. The process of making one cell into two cells of similar size and composition is intricate and complex, like a beautiful machine or a work of art, a song or a dance. On the screen, synchronized animal cells are going through their cell cycle. Several hours of cellular activity reduced to several seconds. The makers of this time-lapse film have used some chemical tricks to help us see two important components of the cell. In blue, DNA, the critical part, chromosomes. In green, a protein called tubulin that aligns the chromosomes and ensures that they get properly distributed to daughter cells. The cell cycle is impressive, but another aspect of living things is equally impressive: development. Multicellular organisms, plants or animals, begin as a single cell. A sperm and an egg, the germ cells, have fused, forming that initial cell, an event called fertilization. After an orchestrated process, repeated cell divisions and progressive specialization, an embryo emerges, then a fetus, and finally, a mature, complex organism with many cell types and functions, including the important ability to make new sperm and new eggs. This time-lapse film reduces a couple of days of frog development to a couple of minutes. First, a frog egg undergoes rapid division to form a large ball of cells. The many cells then follow individual fates. They develop into an organism with a head, a nervous system, and a spine. The eyes form in an oral cavity, and then a beating heart. The embryo becomes a swimming tadpole, and later, an adult frog. You should now be asking yourselves some questions. How do these wondrous things happen? How can a cell know how to go through the cell cycle and divide? And how can the many similar cells in an embryo know how to develop into a complex organism like a tadpole? The answers begin with a startling idea. Cells have information. They have a program. Cells are basically smart. If they were computers, we'd say they have billions of bits. They know how to divide correctly and develop into complex organisms because they have inherited those instructions. What is that information, and where is it located in a cell? What is its chemical form? How do cells interpret and use it? What happens when the information changes? These are among the questions that have driven biologists for over a century. The information resides in chromosomes, in the component of chromosomes we call DNA. DNA is composed of two long interwoven strands, the double helix, chemical chains assembled from four distinct units, colloquially called bases and named A, C, G, and T. The information in DNA is determined by the order of these four bases, just as the meaning of the English language is determined by the way we order and arrange its 26 letters. For this reason, scientists often think of DNA as a text, written with four distinct letters. For tonight's purposes, we'll talk about DNA as a musical score, composed with only those four distinct notes, A, C, G, and T. Over the past half century, we have learned how the cell reads its four notes and what an entire score looks like. We now know something about how a genetic score makes the cell's instruments play together harmoniously to produce the music of the cell. But we are still learning how to interpret all of their DNA. So I'll focus on the part of the score that we understand best. That small fraction, less than 3%, that tells the cell the recipe for its proteins. Proteins are strings of amino acids that carry out much of a cell's work. The score contains instructions to order the amino acids in each protein. In the regions of DNA that encode proteins, three adjacent bases constitute a word, a triplet in musical terms. Each of the 64 possible triplet combinations tells a cell to add one of the 20 kinds of amino acid to the growing end of a protein chain, or to end the chain. Cells can make more than 20,000 different proteins, some at different times, some in different types of cells. Each protein coding region in the DNA score is a gene. These ideas should provoke some further questions. How big is a cell's score? How does the cell copy it to provide a valid score to daughter cells? And how does the cell actually read the score to make proteins? First, we need to appreciate the impressive size of the score. There are about 3 billion notes in human cells. The score is not that much smaller in small creatures like fruit flies and yeast, and it's even pretty large, at least millions of notes, in bacteria. There is so much DNA in a human cell that it would be nearly 6 feet long if it were laid out end to end. To cram so much DNA into tiny cells, it's folded up to form chromosomes. This animation by Drew Barry provides an artful concept of how that might happen. With the assistance of abundant proteins, shown here in blue, the DNA gradually coils up. As one long double strand forms a protein-coated chromosome, you'll see the cartoon version melt into a real chromosome in a dividing cell. For cells to divide, they must duplicate all this DNA, and they must do so very accurately. Watson and Crick made the great discovery that offered insight into how the DNA score can be copied correctly. Well, the double helix is beautiful, but the really profound idea is the pairing of the bases. The A note on one strand of the helix is always paired with a T note on the other, and a C note is always paired with a G. Strands are, in a sense, mirror images. The information in one strand is reflected in the other. So copying one strand will create the reflection, and copying the reflection will recreate the original. And this is brilliant. Let's look more closely at this process of DNA replication by showing more of Drew Barry's cartoons. Here we can see one small region of DNA by being copied by a protein machine composed of the enzymes that make DNA. The proteins are represented as irregular, colorful shapes. The double helix is unzipped to form two single strands, and the single strands are rapidly copied to make more double helices. timing here is realistic. Synthesis of DNA is going on in millions of our own cells at this pace, at this very moment. And in this way, all of the DNA in each of our cells can be copied in only a few hours during one of the phases of the cell division cycle. So how is the information in the DNA notes used to make proteins? The information pathway is called the central dogma of molecular biology. DNA makes RNA, makes protein. First, the order of notes in DNA is transposed into what is, in musical terms, a different key. This process yields a slightly different kind of nucleic acid, called RNA, again with 4 bases. As the film shows, another protein machine, a complex enzyme, finds a place on the DNA to begin its work. Once located on the DNA, it copies one of the strands of the double helix to make what you'll see as a yellow ribbon of RNA. The small yellow objects are the bases that get joined together to form RNA as the enzyme moves along the double helix of DNA. RNA, like DNA, is informative. The order of its bases perfectly matches the order of the corresponding bases in DNA. After a full piece of RNA is made from one gene, it swims through the nucleus and then emerges in the cytoplasm. Each ribbon of RNA in the cytoplasm ends up in the arms of another machine, the protein synthesizing machine called a ribosome, and represented here in blue. The ribosome reads triplets of notes in RNA, making proteins. The red chains that you can see now emerging from the blue ribosome. And it does that by grabbing amino acids, which are represented as red dots on the green carriers. The amino acids are added sequentially to growing chains according to the triplet code. Eventually, red protein chains fold into mature proteins. And the protein you're going to see being folded happens to be globin, the protein in red blood cells that carries oxygen in the blood. Making a protein in this way is like playing one line of sound in the rich music of the cell. When many RNA molecules have been translated to make the many proteins needed by that cell, we can say the DNA score has been interpreted to make a cell's music. We've been talking about a pervasive but simple view of life. The essential features of all living things, regardless of species or of cell type. But of course, life is not simple. It comes in many different forms. This kind of variation is an essential feature of life on Earth. Such marvelous variation was generated, and indeed is still being generated, by changes in DNA, the mutations that drive the evolution of species. We've been talking about general rules, the organization of a cell, the DNA score, the cell cycle, the central dogma, and the way biological instruments copy and read the DNA score. These rules apply to essentially all forms of life: bacteria, plants, insects, mammals like us. It's like knowing that all musicians, classical and jazz, use more or less the same kinds of notation and even the same instruments to make very different kinds of music. And life, like music, is various. Now we must ask, how did such variety arise? Life probably commenced with a unique event 3 or 4 billion years ago. A fortuitous joining of chemicals probably formed a short piece of RNA, able to copy itself. Later, the RNA was wrapped in a membrane to form a primitive cell. From this uncertain beginning, over thousands of millions of years, the multiplicity of life forms we see today was generated. Our understanding of evolution begins really with Charles Darwin and with his less well-known contemporary, Alfred Russel Wallace. Their picture of the origin of species was based on 2 important ideas: variation and selection. First, members of a species produce progeny with varied characteristics. Then the survival of the progeny depends on an ability to thrive and to propagate in their environment. In this way, those that are best adapted are selected. But what is the source of the variation? Here's the part that Darwin did not live long enough to know. The variation is due to changes in DNA, to mutations. Now, we cannot fault Darwin for not knowing about DNA. In the 19th century, no one knew anything about the chemical basis of genetic information or about DNA's base pairs. Recall that cells contain literally billions of bits of information and multiple chromosomes. With that much material to copy, to correct, and to sort into daughter cells, inadvertent errors are inevitable. In other words, when the DNA score is copied so that cells can divide, and when chromosomes are distributed to daughter cells, mistakes will inevitably occur. A chromosome may fail to go to the right daughter cell, or a chromosome might get rearranged, or the wrong base may get inserted in place of the properly paired base. Even if such mistakes are relatively rare, suppose one mispairing every 10 million bases, that would still mean hundreds of errors every time a human cell divides. Most cells know how to correct such errors, but some mistakes will still slip through. These errors, these mutations, will turn up in the daughter cells. Some may dictate an error or a change in the amino acid content of a protein. Others may change the abundance of a protein. Some may have no obvious effects. what is the biological significance of these errors? As in musical mistakes, the context matters. A flub note during a performance may never recur. But if someone incorrectly copies a rare musical score, the mistake is perpetuated. In a similar way, a mistake in an adult cell affects only that single expendable cell. But a mistake in a germ cell, a cell that becomes a sperm or an egg, can affect an entire organism in the next generation. So the mistake can be perpetuated and affect all subsequent generations. Such changes in DNA drive variation and have generated the diversity of living things that Darwin observed and we currently observe. We now have the tools to compare the scores of many species, human and non-human, living and extinct, looking for DNA variations. We can often reconstruct how changes in DNA generated the diversity of living things. Some mutations may have little or no effects. Others may dramatically change the appearance or behavior of an organism. Many factors, geography and food supplies, climate and sex appeal, can then determine which variants will survive, which will thrive, and which will reproduce. This is what Darwin called, and we still call, natural selection. Some varied characteristics may not confer a clear advantage or disadvantage in that battle for survival. Those traits may be tolerated and thus persist. For this reason, even within a single species, a lot of variation in appearance and behavior can be found. And this is especially true among domesticated species. When DNA from each of these dogs is read, the scores look very similar. They differ by no more than 1 base in 1,000. And this is about the same as the difference between dogs or humans that look much more alike. But because the DNA score has 3 billion bases, there are still about 3 million differences. How and to what extent do these differences in DNA cause differences in behavior, in athletic ability, in appearance, in health? How much do genes influence what we become? This is a bit like asking, why does one symphony sound different from another? The notes in the score certainly have a big effect. A symphony by Mozart will always sound very different from one by Shostakovich. But even the same symphony can sound different on different occasions. The conductor, the players, the instruments, and the hall have their effects too. Identical twins can have the same genes, but we can usually tell them apart. The same song sounds different when played by Chet Baker or Miles Davis. Learning the importance of genes, how they exert their effects, and how those effects are modified by environment and behavior is among the great challenges for modern biology. The arts, painting, dance, theater, as well as music, also evolved by variation and selection. Variation in the arts is rooted in the imagination of the artists. The process of selection depends on the approval of those who appreciate art and those who pay for it. Such changes have occurred in portrait art over the last few millennia. We've been thinking about genetic variation and evolution. We've seen that mutation is essential to species diversity, just as stylistic variation is essential to the arts. Without genetic error, there would be no evolution. Without variety, there'd be no development in art, in literature, or in music. Variety is essential to progress. But now we consider a dark side of genetic variation, cancer. Genetic changes can occur in individual cells of a complex organism, during development, after birth, or later in life. The changes sometimes confer an important selective advantage to the mutant cells, an ability to grow more vigorously than normal cells. In this way, mutations, changes in DNA, promote an evolutionary process, variation and selection, among an individual's cells, just as changes in germ cells promote the evolution of species. Recall that all the cells in a human being, all 10 million of them, are descended from a single cell, the fertilized egg. Each of these many cells has a genome, the full complement of hereditary information. And as you've learned, when that DNA is repeatedly reproduced and delivered to daughter cells, during development, during growth, during repair of adult tissues, changes in DNA will inevitably occur. As in evolution, many of these changes in cells are inadvertent. Others may be caused by exposure to mutation-causing agents in tobacco, in food, or sunlight. The mutations vary. Some are subtle alterations in base pairs, some rearrange chromosomes, some rearrange chromosomes by relocation, by deletion, or by duplication. Many mistakes, one-note changes, or larger rearrangements won't change the behavior of cells in any measurable way. Most won't change the protein coding sequence. Other changes could handicap adult cells by making defective proteins, and those cells might simply die and disappear. This won't have any obvious effect on the organs in which they reside, because those organs have hundreds of millions of healthy cells. But some mutations can confer new and disturbing powers on a cell: excessive growth and vitality. These mutations can affect the behavior of cells, especially those epithelial cells that perform specialized functions in the intestine, the breast, the lung, or the cells in our blood and immune systems. Random mutations that enhance cell growth provide a selective advantage, and the outcome of this cellular evolution is a cancer. In this film, the mutant cells, shown in greenish gray, grow inappropriately in a quiescent tissue. The cells violate the normal boundaries of the tissue and form a tumor. They encourage the growth of new blood vessels. The cells may enter the bloodstream, spread to other sites in the body, and invade to grow in those other places. This is called metastasis, and it's ultimately often lethal. These properties are the essence of cancer. Many factors like smoking and lung cancer, viruses and liver cancer, and other things are likely to contribute to cancers like these. Still, we have a unifying idea about how such factors change normal cells into cancer cells. They provoke mutations. Cancers are united by mutations, changes in the DNA score. All the risk factors for cancer contribute to genetic change. But what kinds of mutations actually lead to cancers? Some answers are already known. The most common cancer-causing mutations affect only a few hundred of the 20,000 of our genes, especially those genes that normally govern the growth, the development, and the death of cells. Cancer-causing mutations are good for that cell. They potentiate its growth and its survival. They provide a selective advantage. But they are bad for the organism. The mutant cells can spread, destroy vital organs, and ultimately kill the host. The mutations themselves can be surprisingly subtle. Here is part of a small gene called RAS, a gene that plays critical roles in many cells, in cell growth and division, cell shape and mobility, and in cell death. A single altered base pair, like the one shown here, is found in about a third of human lung cancers, about a half of human colon cancers, and nearly all of human pancreas cancers. A change in this note is critical. It alters only one amino acid in the RAS protein. But that change alters the protein at a critical site and makes the protein resistant to its normal controls. So now it persistently tells the cell to change its shape, to avoid death, and most importantly, to grow, and grow. So the cells divide too often. The extra cells overgrow their neighbors, and the cells often spread elsewhere. Other kinds of genetic errors in cancer cells are less subtle. At the top of this image are the chromosomes of a normal cell, painted chemically with colors to show each of the 23 pairs. Below are selected chromosomes from cancer cells. They are abnormal in number and composition, and some look like harlequin clowns. You might expect cells with such mangled chromosomes to die, and many probably do. But some of these cells not only survive, they grow better than the cells from which they arose. They may grow better because they have more copies of genes that tell them to grow, or they may have rearranged genes, and proteins made by those novel or distorted genes may send a persistent signal, again, to grow. Cancer is not just a fascinating biological phenomenon. It's also a dreaded disease, one of the world's leading killers, affecting all of us, directly or indirectly. So it's now important to ask, does knowledge about the alterations in a cancer cell score help cancer patients and the doctors who treat them? Three strategies are commonly used to treat cancers today. A surgeon can often remove all detectable cancer, giving the best chances for a complete cure. To do this, the surgeon doesn't need to know about genes and mutations. Cancer doctors can use toxic chemicals or radiation to damage or to kill cancer cells. But again, they don't absolutely need to know which specific notes have been changed in the genetic score of the cancer cells when they use these tools. But some new therapies are focused on the precise mutations in cancer cells. The idea is simple: to use precisely targeted drugs to counter the action of mutant cancer-causing genes. Ideally, this is a chemical that spares normal cells and will kill a cancer cell by blocking a rogue protein synthesized from the mutant score of a cancer gene. Chronic myeloid leukemia, a relatively common leukemia of adults, is the most successful example to date of this important new strategy. The cells in this leukemia are driven by a rearranged chromosome, called the Philadelphia chromosome, discovered over 40 years ago. It's produced by the inadvertent breaks in chromosomes. When pieces of two broken chromosomes fuse, and they form the basis for this leukemia, they have formed a new hybrid gene. The gene makes an enzyme that's abnormally active and can't be shut off by the cell's usual controls. Happily, about a dozen years ago, a drug, a small chemical called Gleevec, was discovered that can inhibit that supercharged enzyme by binding to it. When patients were first given this drug, roughly a decade ago, especially when given it early in the disease, nearly all the leukemic cells died. In those patients, the blood and bone marrow resumed their normal appearance. And now thousands of patients on this drug are doing fine as long as the drug continues to suppress the miscreant enzyme. So, what have we learned about cancer tonight? Cancer reflects some basic principles in biology. Cells evolve like species by variation and selection. Cancer reminds us that change is inevitable in biology and essential for the diversity of life. But it's also threatening. Genetic change can make cells cancerous. We have also seen an inspiring new kind of cancer treatment, Gleevec, a drug that precisely blocks the change that made a cell leukemic. But this strategy has not yet worked so well for more common cancers. For some lung cancers, we have Gleevec-like drugs, but the cancers soon become resistant to them. For the many cancers with RAS mutations, we haven't even found such specific drugs. Still, there are reasons for optimism. Our new understanding of cancer as a disease of genes. New drugs that overcome resistance to the older drugs. New ideas for countering mutations, even RAS mutations. And most of all, determined scientists and clinicians, passionate advocates for patients. Tonight, we have aimed to entertain and instruct. We have also tried to show how sciences and arts can perform together and borrow ideas from each other. Jacob and I believe we both work in worlds that demand creativity and observation. Worlds in which knowledge, methods, and style evolve. And worlds that matter, we hope, to all of you. Please thank Turu Dodo on the piano. Brian Woodruff on the drums. Kevin Thomas on the bass. Erica von Kleist, alto and soprano saxes, and the flute. Jacob Varmus, trumpet, cornet, composer, and musical director. Thank you all for coming tonight.
If anyone has any questions or would like to just say hello, Dr. Varmus and the musicians will be here, and feel free to come on down and say hello or chat a bit if you'd like. And thanks so much for coming.